Welcome to the Fundamentals of Digital Imaging lecture. This is the introductory lecture of the FIJI for Beginners workshop. In this lecture, we will cover the digital image, some image acquisition tips, basic rules of digital images, and image presentation guidelines. This will be followed in the workshop by some hands-on applications on how to import, manipulate, annotate, and present microscopy data. So let's get started with the digital image. Microscopy data are digital images. This is made up of pixels. And so although we see the data as an image, it's essentially a numerical array of intensity values. So it's essentially an Excel spreadsheet. Each pixel has some certain characteristics. So first of all, there's a representative size of the sample. In this example, the pixel is 100 nanometers in X and Y directions. And it also has an intensity value here, in this case, 108 grayscale values. So this is given by the bit depth of the image, and the size of the pixel is given by the sampling resolution. And we'll cover both of these topics in the coming slides. So first of all, bit depth. Essentially, this gives you the number of grayscale values within the image. So a 1-bit image is 2 to the power of 1. That gives you 2 gray values, so either black or white within the image. We often term this as a binary image, and this is used quite a bit in image quantification. If we go to a 2-bit image, that's 2 to the power of 2, which gives you 4 grayscale values, and so on and so on. Why is this important? So if we look at these two examples of a 4x2 array of pixels, one is acquired on an 8-bit scale, so between 0 and 255 grayscale values. If you look at the 2 pixels here, they essentially have identical intensities. Whereas data which is acquired at a 16-bit setting, we can actually now see there is different intensities between these 2 pixels. This become very, very important downstream when we're doing image quantification to allow us to separate out different objects within the image. This is another example showing the effect of bit depth. So here we've taken a microscope image and we've turned them into different bit depths. So here is a 1-bit image, so either 0 intensity or maximum intensity. And as we increase up the bit depth, you can see we can get more and more information out of the image. Once we go beyond 8-bit, it probably won't make any visual difference because the projectors and the screens that we use often work to an 8-bit setting. However, if we look, as I mentioned, quantifiably, if we draw a line there and look at the intensity across that line, you can see as we go up in the bit depth, we get more and more information about what's happening across of the intensity values across that line, across our sample. This is some examples, some microscopy examples of bit depth. So as I mentioned, a 1-bit image gives you 2 gray values. That's used for binary masks for image analysis. And if you attend the FIJI for quantification workshop, we'll use that quite a bit. A standard setting is 8-bit. These are used on scientific cameras and also on confocal detectors. With the cameras, they tend to be, you will buy an 8-bit camera or 12-bit camera. On the confocal side, we can reassign intensity values to 8, 12, sometimes 16 bits. So these are some examples of bump equipment that have different bit depths associated with them. The real take home from this is if you're imaging on the microscope just for qualitative purposes, just for visualization, then 8-bit is probably fine for any presentations that you will be giving. If you do want to quantify the data, then we suggest you go to a higher bit depth if possible. Certainly on a confocal system, you can vary this up to 12 or 16 bits if you can. That will help you with any downstream analysis. Okay, so that's the digital image and a couple of things to think about there. Next, we'll go through image acquisition and how to set up the microscope to take a good quality microscope image. So often one of the things we look at first here is something called the intensity graph. I've shown a couple of examples of this here. So on the x-axis, we have the intensity scale. So this is an 8-bit image. It's going from 0 up to 255 grayscale values. So at the 0 level, we're looking at low level intensity. And as we go up, we're looking up to the maximum dynamic range of the detector. On the y-axis, we can look at the number of pixels that have that intensity. So just by looking at the intensity graph, we can see in this example that the majority of the pixels have very low intensity. So this is probably an image with a lot of background in it. Whereas if you look at this example down here, the intensity values are a lot more spread across the intensity range. So this is probably an image of something like a piece of tissue where there's sample across the entire image. The other thing to note here is that this is not on an 8-bit scale. This is running from 8 to 65,000 grayscale values. One important thing, and we'll cover this a little bit later on, is in terms of any modifications that you're making, but also how you present the data. So here you can see there's this line across here. We can drag each of these little squares. So the adjustments we'll be making normally for ethical purposes are kind of min and max adjustments. Here we probably wouldn't make any changes because the data covers the entire dynamic range. One problem here we have is that actually we're saturating the detector. So we've got saturated pixels here that we can't actually quantify. One way of looking at this is to use a specialist lookup table, which will identify any of these saturated pixels in red. And we can do this at the time of acquisition of the data to make sure that we're not saturating any pixels. So in this case, we could decrease the excitation light going onto the sample, or minimize the exposure time if we were on a camera system, or decrease the gain if we were on a confocal system. Ideally, you want to use as much of the dynamic range as possible without hitting saturation at the top of the intensity graph. One other thing we need to look at when we're actually acquiring the data is the sampling. So if you remember, I mentioned that your pixel has a relative dimension that corresponds to the sample that you've imaged. And what we're doing when we're taking a digital image is we're creating what's called a raster image. So essentially, what we've captured is divided up into pixels. This is very different to if you create something in something like Photoshop, where the image is presented as what's called a vector. That essentially means each of these curves are associated with a mathematical function, so you can zoom in and you won't get any increase in pixelization. Whereas if you zoom in on a digital image, that pixel should grow as you zoom in. You can't change the sampling rate after acquisition. That means we need to think about our sampling rate while we're acquiring the data. So in this little task, we're going to have this image of a blue duck, and we're going to convert that into a digital image. And say your research question is that you want to identify and count the number of ducks within an image. So how often do we need to, or how much do we need to break up that structure that we're interested in into pixels? So if we do that 3 by 3 and we look at the image here, can we really say that we're certain that this is the representation of a blue duck? Probably not. By the time you get to 5 by 5, it's probably better. You're getting closer. By the time you're at 10 by 10, then you're probably happier. If your research question, though, was to count the number of eyes that you could see on the duck, then you might need to go to something like 20 by 20 or even up to 300. The key thing here is to try and get away with the smallest number of pixels within the image to get the most relevant information. So especially on a confocal system where you're scanning across to build up your image, the more pixels you have, the slower your image will take to acquire. By the time you get to 300 by 300 pixels, you've probably oversampled in terms of what you actually need. Fortunately, there is some mathematics behind this, so it's called the Nyquist-Shannon sampling rate. And it says here, in order to convert an analog signal, in this case, the blue wavy line, into a digital signal, in this case, the red line, the signal needs to be sampled at least 2.3 times at the half cycle. So we've got an image here which shows you this. So we've got this wave, which is our analog signal, and the red line is the digital representation. So by the time you get to 2.3 times, it's not perfect, but it's a pretty good representation of your analog signal. Anything below that would be considered undersampling. Anything above that, potentially oversampling. Fortunately, there are calculators which will do this for you. So this is a very good one. And essentially, the pixel dimensions you need are your theoretical resolution divided by that 2.3 value. On most confocal systems now, you will find an optimal or a Nyquist button, which will do this calculation for you. And the table here shows some examples. So if you're on a 63 or a 100x objective with an NA of 1.4, which gives you your resolution of that objective, then ideally in the kind of green wavelengths, you want to have pixel dimensions at about 60 nanometers. Now, the really important thing, and I would stress this, is you only need a 60 nanometer pixel size if you're trying to get the best possible image quality out of your microscope. If you're trying to count, say, nuclei or cells, and your cells are much, much larger, then obviously you do not need pixels that are 60 nanometers in size. So again, that's something to think about at the start of your experiment. What is it that I want to quantify at the end? And therefore you set your acquisition settings accordingly. The other thing we're trying to do is to get the best representative image of what we can see down the eyepiece. So what we want to avoid is if your sample prep hasn't worked, you shouldn't get a very good image. So if you put crap in, you should get crap out. The microscope isn't a magic box. 90% of the battle in microscopy is sample prep. So if you can get that right, then the imaging should be easy. What we want to be careful of is if our sample prep hasn't worked, if we change different parameters, then we can actually get different results from our data. When we are setting up these experiments, then we suggest you don't use offset on confocal. So that's a way of clipping the low intensity values. We would set your acquisition settings using positive and negative control. So something that you know is going to be the brightest, something you know is going to likely to be the dimmest. You should be trying to acquire with the same settings between samples, especially on data sets that you want to directly compare. And we also suggest you run a pilot analysis before and during your imaging as well, just to make sure you've done the entire workflow at least once before embarking on a large project. Okay, you've acquired your data, so now what should you be doing with those images that you've taken? So the first thing in, hopefully, is fairly obvious, is to save, but save it as the raw file extension. So there are some examples here from different vendors. If you can save that, it not just saves the image data, but it also saves the metadata as well. So if a reviewer or your supervisor asks you, how did you acquire the data on a given day, you can go back to this file, open up the metadata, and it will tell you what lasers and what the microscope settings were, what your objective was, that type of thing. The other nice thing that allows you to do often is to reapply those settings again. So you're imaging the exact same way another day as you were the previous day. After that, we ask that you export your data out. So you should always keep the raw data file as your kind of ground truth. Any modifications you should be doing to exported data, we would suggest that you should do that as a TIFF file. And again, that's because it keeps that raw information. If you use some of these other formats, so JPEG, GIF, or PNG files, you will find that they are compressed very often in what's called a lossy manner, so you're actually losing information. We'd suggest that if you're going to use these, they're very useful for lab presentations or putting into your thesis, but any quantitative work should be done on a TIFF file. Okay, as I mentioned earlier on, any modifications should be done on a copy. So never process or modify the raw data. And as you make modifications, it's always a good idea to either say in your lab book what you've done, or more directly, change the file name as you've made the modifications so you know what's happened at each step of the way. Another key thing is to back that data up. So it's a requirement of your funding body that you safely do that. So you should be doubly backing up your data. So if there is a problem on one copy of it, then you have a backup copy elsewhere to be able to do that. That should be pretty easy now, especially at the university. They have data redundancy on your research drives. You also have access to cloud services as well, which will allow you to do this. The other really key thing that we find in our platform is please do not use the microscope computer to store your data. So there are often lots and lots of people who have access to that. We have had instances in the past where people have accidentally deleted people's data, and that data is gone if you haven't already backed that up. So get in the habits of using Data Mover, which is what we use in our platform, to get that data off the computer as quickly as possible. And then we'd ask that you delete that from the system next time that you're on the microscope. Okay, we touched on this a little bit at the start, but again, although we're creating an image, it's essentially an Excel spreadsheet of numbers, which means it's scientific data. So any modifications you're making to that image can also be seen as being data manipulation. And it's not good enough nowadays to say, oops, I wasn't aware of it, but I thought it would be okay. So what we're going to do over the next few slides is show a few examples of what not to do, and hopefully that'll highlight how you should be behaving. So over the next few slides, we'll have an original image on the left-hand side and then a manipulated image on the right-hand side. In this example, this is an electron microscope image where they have immunolabeled a certain structure. And normally the way you detect this in an electron microscope is a nanogold, which is attached to one of your antibodies, which is highly absorbent, absorbs the electron beam, and should be fairly visible. So in the manipulated image, you can see that they've increased the contrast, increased the darkness on these objects here. I guess they presume that these are the immunogold, certainly a lot darker than in the original image. They also have this speck here, which they've gone in and taken out of the image as well. So hopefully you guys can understand that that's not really ethical. They're making modifications to specific parts of the image. If this hasn't worked, then they should go back and do the sample preparation or the sample acquisition again. In this case, this is a fluorescence image. So here we've got a bunch of cells, and in the manipulated image, if we go in and do a simple contrast adjustment, we can actually see that it looks like these cells here on the left and these on the bottom right have a very different background to those from the rest in the image. It's very possible that these have actually been cut and pasted in from another image, and yet you wouldn't be able to see that very easily just from the raw data. The journal editors now are very aware of these types of modifications, so do lots of tests of the data that's being submitted to publications. Again, you don't want to be doing that. In this example, there's a fold in the original image which has been taken out in the manipulated image. Again, you're making a specific modification to a single feature within the image. What could you do? So either live with it if it's not too bad, if what you're interested in is over here and it's not interpreting the analysis of the image, then just live with it. Or if what you're interested in is over here, you can just crop it out so it's not within the image anymore. Or if it is important, you should have a tissue block, which you can go back to, take another section, try and find one without a fold in it, and use that for your final figure. In this example, we've got a shading problem. So this microscope hasn't been set up properly, and there's a bit of uneven illumination in this bottom right-hand corner, which has been corrected post-acquisition. So my argument here would be that if you can see that at the time of acquisition, you should realign the microscope, make sure your illumination is even, and then you don't need to make the modification post-acquisition. However, this is mostly acceptable only if no changes actually occurred in the actual tissue, so it's just the background region, and a statement is made in the figure legend. In general, any modifications you make to your data, so even brightness and contrast adjustments, you should be making that statement in the figure legend or the materials and methods. In this example, you can see that the uneven illumination has been cleaned up, and some of the tissue around the outside has been erased as well. This is deemed not acceptable due to a large degree of background alteration. So again, not appropriate. So what we can do is make modifications to the whole image. So in this example, we've got a 3-channel microscope image. And what's happened here is it's been acquired, but we haven't used the full dynamic range of the detector, so the intensity values don't go all the way out to the maximum. And as a result, the image isn't the brightest. Sometimes can be hard to maybe visualize in a presentation or in your thesis or something like that. So in this case, we can change the maximum value down to the point where we start to hit some of the intensity values. And we can use that lookup table that I mentioned before, where if we hit the data, we'll start to saturate those pixels, and they'll come up as red. So in this case, we've brought it down to a level where it's made the image brighter, but we're not saturating any of the pixels. However, if we go too far, as you can see here, we've got a bunch of different signals which are hitting saturation. And what you find in the final image is actually, if you compare this panel against this panel, some of these structures look a lot larger than they actually are. And again, this can affect the interpretation of the data. So you can change the brightness and contrast up until a point where you hit the data, but not beyond it. Okay, in this case, we've got a fluorescence microscope image. This person's obviously attended my talks in the past because they've got their sample, but they've also got a positive control and a negative control as well. However, again, if we go in and do a simple contrast adjustment, we can see that the backgrounds between the sample, the positive, and the negative control are very different. Now, this is concerning. That suggests that either the data has been acquired different ways, or they have been handled different ways post-acquisition. So it's likely they've not been processed identically. And again, that's not something that we want to do. Okay, once we've gone through our data that we've acquired, the last thing is, how should we present it? And the FIJI for Beginners workshop will go through a lot of these steps in terms of how to present that data, but there's a few things to think about here. So the first thing is the data presentation guidelines. So each of the journals has their own instructions to authors. This is a copy from Cell from a few years ago. And again, this just reiterates what we've already covered in this talk. So any alteration should be applied to the entire image and not specific parts within an image. Any alteration should be explained within the figure legend. If you are trying to compare data, then it should be acquired the same way within the same experiment. And this is journal specific, but individual images should not be used in multiple figures unless the figure describes a different aspect of the same experiment. So as I say, each journal will have their own instructions for authors. It's worth, if you're a student, looking at your lab and see what journals your lab publishes in and checking what the instructions to authors are for those journals. A couple of other things for you to just be aware of are things like color sensitivity and color blindness, and we'll cover some of those examples later on in this talk, and then also printing issues as well. Again, we'll cover that in the next few slides. So when it comes to color, most fluorescence microscope detectors are actually monochrome, so they're not really detecting color. Normally we collect on a monochrome detector. It has some kind of filter in the front, which is blocking out certain wavelengths of light. That means the images tend to be grayscale, so it's just an intensity range, right? So what we're doing by adding color is using something called a lookup table. Now, these are three examples of the most commonly used lookup tables. So we've got the blue, green, and red lookup table on an 8-bit range from 0 to 255 grayscale values. And this is a linear range between black and bright blue in the blue case, black and bright green in the green case. Okay, so this is the exact same microscope image presented in the blue lookup table, the green lookup table, and the red lookup table. And if we zoom into one specific part of that, I would argue that most people would be able to see in the green, with the green lookup table, more detail than they can in the blue and the red. And that goes to how our eyes interpret color. So we see very well in the green and red wavelengths, not so well in the blue wavelengths. So this is a very simple representation showing you, depending on what color you've assigned to each of your channels, that can affect the interpretation of the data from those channels. The other thing that we're looking at when we're looking on a screen or on our phone is we're looking at what's called the red, green, blue color palette. So in this case, it's an additive color palette. So we're adding intensity of green, red, and blue. That's how the images are created on our screens. So if we've got full red, no green, no blue, we see red. Similarly with full green, we see green. And once you start to mix in the different red, green, and blue color palettes, if you add red and green together, you get yellow. Red and blue gives you magenta, etc., etc. So that's fine in the digital world, but when we come to the printing world, it often works in what's called the CMYK color palette, which is works very, very differently. It's a subtractive color palette. And this might explain why, if you've ever tried to create a figure, it looks great on your screen, but when you come to print it, sometimes maybe the greens are a little bit washed out. So this is another representation of that. The triangle here shows the red, green, blue color palettes, and this kind of weird shape shows the CMYK color palette. So again, your greens look great on the screen, but when you come to print them out, they're a bit washed out. And this is why you get that effect. That means, again, that we need to be very careful when we're creating panels. So in this case, I've got a blue, green, red panel here with a merge. Again, we'll come to it, but this green and red combination is not the best. In terms of making the channels equally interpretable, I would argue that you could potentially use a grayscale lookup table that allows you to see all of the information irrespective of the color that you're using to present that. You can still have a merge image on the final panel, but yeah, I would suggest maybe avoiding the green and red, and we'll cover that in a couple of slides' time. One other possibility is to use a grayscale lookup table, but use an inverted lookup table. So here, our background intensities are white, and our foreground, our higher intensities are staying in black. And this can certainly save you ink if you're trying to print these out, but also can help with visualization of the data as well. And then finally, I mentioned the red, green, blue is not a good combination. Certainly the red and the green is a common color blindness. So that's probably one to be avoided. There are other combinations that work reasonably well. So things like cyan, yellow, and red, cyan, yellow, and magenta, or blue, green, and magenta as well. So again, use the individual channel panels as grayscale, but maybe use something like blue, green, red as your merge channel.